Technology is enabling new methods of exploring vaccine candidates for trial, but there are already a few tried and tested ways to make them. In all of them, scientists try to stimulate the body's immune system to combat invasive pathogens. That's commonly done by creating something so similar to the pathogen that the body begins to create antibodies to fight off the real thing. The most common way of doing this is to make what's called attenuated vaccines, those that are made of weaker strains of the actual pathogen. Reared on animal cells outside of human bodies, some flu vaccines are cultured on chicken eggs. They are then extracted and injected in a single tiny dose. Vaccines for measles and tuberculosis are created in this way. Inactivated vaccines, on the other hand, are derived from identifying the active proteins in a virus that enables them to invade human cells. That's done by taking dead samples of the pathogen and studying their genetic makeup so that scientists can replicate them in a group. When injected into a human, the body gets to work again constructing the necessary antibodies. Often such vaccines require multiple doses over time, including those to protect against such diseases as rabies and polio. Then there are nucleotide-based vaccines, which seek to replicate the genetic material, DNA and RNA, of a virus. This method stimulates the body's own creation of this viral material in order for it to produce the necessary antibodies. Vaccines need lots of tests. Most of the vaccines we rely on today took between 5 and 15 years to perfect. A DNA-based vaccine for Zika virus, which was declared a public health emergency by the World Health Organization in 2016, was ready for clinical trials 7 months after it was designed, but that is unusual. There are already at least 35 companies and academic institutions racing to make a COVID-19 vaccine, with at least 4 candidates in the animal testing phase. And one will enter human trials soon. But that's just one hurdle cleared. As Anthony Fauci, the director of the National Institute of Allergy and Infectious Diseases in the US, said, "A vaccine that you make and start testing in a year is not a vaccine that's deployable. It will take a year to a year and a half, no matter how fast you go. Vaccines must be rigorously tested to ensure they not only work but will not cause other dangerous side effects." The trial methodology consists of 3 phases. 1. Testing on a small number of healthy adults. 2. Testing on a larger number of adults in an area where the disease has spread. 3. Testing on thousands of people in an area where the disease has spread. Each of these steps can last between 6 and 8 months, but even if the vaccine candidate gets that far, many are abandoned or fail before then. They must then be studied by regulators before approval is granted.